Everybody, and will first thank the organizer for inviting me here, particularly Chiara and Umberto Veronesi, to whom I owe a lot of my career and the possibility that he gave me and gave us to build up really academic and independent science here in Italy. Okay, I'll switch to Italian, and I apologize with the non-Italian speaking colleagues for that. Quello che mi è stato chiesto di...
I've been asked to discuss, as Professor Testa mentioned, to discuss what our ability and what our possibility of digitize, of measure the effects of the environment on the individuals, as far as the possibility that this effect may contribute to maintain health or may contribute to causing diseases or risk of developing diseases. And this is what I've been studying. Today we will be discussing the DNA and how DNA variations and modifications may cause diseases or the risk of developing diseases. This is connected with the very fact that there are DNA variants called alleles, which are causing differences between individuals and which may determine a different fate for the single individuals as far as the risk of developing disease is concerned. We are discussing the 3 billion of nucleotides per genome, which means that we have 99.99% of our sequences of the genome are just identical, but there are small differences between us, amongst ourselves, which is even less than 0.5%, which nevertheless is equal to 30 million nucleotides. So we differ for the 30 positions of DNA, which by themselves may have an impact on health and the possibility of developing diseases. The possibility of recognizing some of these variants determines what we discussed yesterday, i.e. the individual risk of developing a disease should we be able to identify it. In concrete terms, I would like to make an example. We have genes BRC1 or 2, which if these genes are present in a determined form, may increase by 30% the risk of developing a tumor in women carrying that variant. The number of tumors or the percentage of tumors which is genetically determined, which in a way or another is written in our DNA, amounts to 20%. So not all of us, not all the diseases. It is recognized that the environment or the environmental modifications caused by each one of us, i.e. the lifestyle and the environment can be responsible for health or the increase in the possibility of developing diseases. It is not easy to determine the percentage which causes or the percentage affecting us, because we cannot be certain of the causes and the changes. The WHO determines that about 30% of all diseases are associated to known environmental factors. Should these environmental factors be removed, then the incidence of that disease could be reduced, and 30% we are discussing here all kind of diseases, infectious diseases, which are easily correlated with the environment, but we are also considering cancer and neurodegenerative diseases and cardiovascular diseases. In the case of tumors, we estimate that 60% of all tumors are caused by environmental causes or by lifestyle choices. Once again, these are percentages to be accepted with a certain caution, because in most cases the real causes are not known. Let's consider instead a situation which epidemiologically is known, is characterized, a case of environmental origin. Let's consider, therefore, obesity. We have a very potent and powerful and strong epidemiological studies. The last one, or the latest, was published 1 year ago and studied 500,000 people. So these studies have shown that there is no doubt about the fact that obesity increases the risk of dying of multiple cancers. Here we have a list of different cancers with an increased risk of dying in the obese patient, from 1 to 7 times more possibility of developing a cancer in certain cases. And the risk of death associated to the presence of the tumor has two components. The behavior tumor is more severe in obese patients, and the second one is the incidence of tumors, i.e. the probability of developing a tumor is higher in obese individuals. But the problem is simple. We do not know how to recognize those obese individuals who will develop a tumor. So there is no possibility of recognizing the individual. In order to be able to do so, in order to be able to determine who will develop a tumor because of that environmental impact, we should be able to measure the effect of the environment on that individual, and should we be able to determine a mechanistic cause-effect relationship between the environment and the tumor. This possibility of measuring the effect of the environment on a living organism presupposes two important factors. First of all, that the environment has an effect on the individual, and that this effect is stable over time, is an effect which will modify the behavior of the individual and which determines a stable variable, a stable change which can be measured. And this could be possible if we had a code for measuring the exposure of the individual to the environment. In other words, we could measure that code, and should we be able to measure that code, we could modify the paradigm without communicating to the whole population the risk, but by discussing with the single individual the risk he or she is exposed to. This, of course, is based on certain phenomena we have been knowing for decades, but which are reconsidered because we now know the code. First of all, cells have a memory. Organisms have a memory. I would like to make an example. For example, if you take these cells in our tissues, these cells are different one from the other. A neuron is different from a skin cell. But the DNA of these two cells in the same organism is just the same. We have heard that there are small modifications in the DNA within each one of us, which are absolutely not involved in the differentiation of the cells in each organism. But this differentiation takes place in an early stage, in the embryo. which are taught who they are, and this datum, this information survives throughout the life of the cell. So cells have the memory of the instruction they have been given from the environment outside the cell itself. It's therefore the memory which is not a genetic kind of memory. It's not present in the DNA. Now I move from cells to organisms. Organisms, is that clock working well? No, sorry, I was checking my time. So organisms can adjust to environmental changes. When we speak about adaptation of the organism to environmental changes, now we step into the world of evolution. Evolution is, and the evolutionary theory is based on the theory of the adaptation of the organism to the environment, always according to Darwin, and then to gene adaptation. So adaptation to the environment takes place through natural selection and through the competition of the individuals who have specific benefits vis-à-vis survival or because of survival. The DNA varies in a random fashion, and the environment selects those variations which may increase the individual probability to survive in the changing environment. This brings about an enhancement, an enrichment of that kind of genes, and this is at the very basis of speciation. But that's not all. That's not it, because we can intuitively understand that the environment changes rapidly during the life of an individual. For example, if we consider the life of a human being, it is quite clear that the environment changes radically throughout that life. Suffice to consider the seasons or the volcano eruption or other things which may radically change the environment, thus imposing the necessity for the individual to adapt to those changes. Natural selection by itself, as we have experienced it, cannot comply with these changes, because the time scale it applies is not compatible with these changes. So these changes call for the adaptation of the individual, not the adaptation of the species. Therefore, the human beings and the individuals and the physiological system can respond and adapt to new changes in real time. We have tens of examples, some of which were described by Darwin himself, even though they were not included in his theory. For example, insects, they change their color according to the leaf they stand on. And this change, this color, remains as long as this insect stays on that leaf, or leaves changing shape according to the water content or chemical substances present in the organism. But another thing which is more complex is that these changes, which are caused or are a response to the environment, these changes may be transmitted through the generations. This is normally called transgenerational epigenetics. That means that experiences of the parents or changes brought about by the environment in these individuals may be transmitted down the line, down to 3 or 4 generations without any changes in the DNA. This may be brought about by exposures to the environment, and they may take place in the uterus during the embryo and the fetus development or in the early stage of life. In this case, too, we have a large amount of data taken from epistemological studies in the humans or by studies on plants and animals. I would like to mention a couple of these studies. The first one concerns the exposure of the fetus and the possibility that this exposure may be turned into a memory which is transmitted down to the third and fourth generation of adults to be born. A well-known example is that of children of mothers exposed to the Dutch famine in 1944. In the women where these exposures took place during the last trimester of pregnancy or the first few months of life, for these women, the children were less obese than the controls, or i.e. women who had not been exposed to the Dutch famine, whereas the data changed if the exposure had taken place during the first trimester of pregnancy. So the intake of food is very important in terms of the possibility of developing obesity. And this can be seen down to the second generation of children. The same applies to early in-life exposures. But all this applies to the period before puberty. In another historical study studied the variation in the food supply during the early years of paternal grandparents. This has been seen to be associated with variation in the mortality rate, especially in death caused by diabetes, down to the third generation. And these are examples of adaptation to the environment, which, if read on the basis of the negative impact on the environment, are all effects of the effect that the environment has by changing the behavior. And this causes the development of memory in the cells and individuals. And this memory can be passed down to future generations. And these evidences, which are very numerous, have been left on the margin of science because the mechanism was not well known. But now they can be reproduced in labs, and we have a large number of models which study this inheritance, which is not associated with DNA variations. Just one example to share with you the fact that these environmental models are sound and can be studied in worms, for example. It is possible to have viral resistance which is developed in worm. This resistance may be passed down to the third generation, to the offsprings. This resistance is due to the fact that a specific molecule, a small RNA, is developed in the worm. It is possible to measure the transfer of these small RNA down to the future generations and to detect resistance in the offspring, even when the DNA generating that small RNA is not present, is absent, and also when all the machinery bringing about the production of RNA is absent. We have a very simple model measuring the non-genetic inheritance, showing that this is the case, and this is happening in the absence of the RNA. This is a very difficult experiment, but this shows that there are modifications which are associated with the adaptation to environmental changes and to cell speciations, but which are associated with the memory and not associated with the DNA modifications, and can be inherited through the cells or through the generations. But our way of approaching this phenomena and our possibility of using the basis to our benefit to control health and diseases has been made possible by understanding the code of this non-genetic inheritance. And the code of this non-genetic inheritance is to be found in the chromatin, which forms with the genome the epigenome. The chromatin is a set of histonic proteins associated with other proteins, wrapping up the DNA. The DNA is wrapped up in proteins, and proteins are collectively called chromatin, and they define the epigenome. Little by little, we are learning that the lifestyle, the environment, is decodified into signals which modify the chromatin. These protein wrapping up the DNA and influence the activity of DNA also determine the phenotypes which are environment-induced and which are phenotype to adapt to the environment or to maintain the memory of this adaptation to the environment. Let's start to see what are the molecular mechanisms allowing for this transduction of the signal coming to the environment into behaviors. The first point, we have two aspects in this mechanism. The first aspect of this mechanism is that these signals coming from the environment do determine some markers at the level of these proteins which are stable, and this is what we can measure. Because what happens? It happens that these signals modify the activity of certain nuclear factors which are connected to the DNA and modify the chromatin. These small dots, colored dots, are environmental effects, and they are very numerous. In this slide, you can see the minimum functional entity of chromatin. Here you have histones with these spots where you put the flag, and this is the result of the environmental stimulus. Here you can see 5 different flags with post-translational modifications, which may happen in different positions, in different parts. We know that at least 30 possible modifications, 30 different flags, which may be found in different places. This is a code with hundreds of letters, and it's a combination of words, because the environment sends different signals, and this code brings about and determines changes which depends on the individual experience, because each and every one has different lifestyles or different experiences of environment. So each one of us has its own epigenome. We have our own epigenome telling the story of our interaction with the environment. And we have hundreds of epigenome for each single individual, because each cell has its own epigenome, preserving its identity. So now you can consider the complexity scale. Yesterday we discussed the complexity determined of the genetic code, and with the 4-letter code. But here we have hundreds of genome for each individual with a code which is probably made up of 30, 40 different letters. So the complexity is huge. It's an unprecedented complexity as far as we know, and especially if compared to genomics. And we are starting to read it because over the last 4 or 5 years, we have developed technologies which have enabled us to read these epigenomes, and now we can read each single curve, each single deposit, each single curve. And here we have just a limited number of bases of epigenomes. You can imagine the complexity of it all. Here we have a consortium which has been set up recently, and over the next few years, this consortium has been given the task of mapping 10,000 epigenomes. Well, the complexity of an epigenome is much higher than that of a genome. But 15 years after the mapping of the genome, the costs have collapsed, and the technology allows us to act much faster. So the consortium is generating epigenomes on a continuous basis. So we are starting to develop the picture of the organization of our history of interaction with the environment. I'm running late, so I would like to tackle the second important aspect. This modification in the Mp genomes, these signals are introduced during the processing of these signals coming from the environment, and they are introduced by enzymes, chromatin modificating enzymes, and they are growing in number, as you can see. The fact that these are enzymes offer us the great possibility of modifying them through drugs. We have learned, with some difficulties, how to generate drugs to modify the enzymes. Generally speaking, we could generate drugs which interact with the enzymes which are involved in the transformation of the environmental signals into an epigenomic code. And we have some of these drugs. So this is a demethylase inhibitor drug, and some of these drugs do work. In a couple of minutes, I would like to show you how these drugs may be used by exploiting the ability of the diseased cells, of the tumor cells, to adjust, to adapt to a new environment. Just in 1 minute, Giuseppe, I will close in 1 minute. This is the effect of the reduction of the calorie intake on tumors. You know that if you reduce the calorie intake, you have a positive effect on the duration of life, but also on some tumors. So this is an experiment carried out on an animal. You can see that animals on a standard light, if challenged with leukemia, these animals die at a certain point in time. But if these animals have a reduced calorie intake diet, then the survival rate becomes higher. You prolong the life. So the reduction of the calorie intake reduces the speed at which leukemia develops. But if we consider it, there is a slowing down in the developing of the leukemia, but it's not the cure. You can see in the red curve that in the leukemia in animals with a low calorie intake diet has a slowdown. The leukemia slowed down. There's leukemia which slows down, but then there is a picking up again. So there is no eradication of leukemia. If you study that point in time, when the diet is reduced, that is an environmental change, you can see that there is no change in the DNA, but there is a metabolic adaptation which takes place here at this very point. So the cell learns how to live in that environment with a low intake of calories. The metabolism is changed, but then there is a picking up again of the disease. If you study the epigenome, the chromatin, you may be able to understand which are the enzymes mediating this adaptation response, because this is an adaptation response which is mediated by modifications in the epigenome and in the chromatin. And we have succeeded in identifying this demethylase lysine, this enzyme. If you inhibit this lysine demethylase, you have the cure for leukemia. We are saying that even the tumor cells, the diseased affected cells, are adapting to the environment, and we can benefit from this situation by exposing the tumor cell to dramatic changes in the environment, reduction of the change in the diet, and the tumor cell must adapt, and it's very good at adapting, and it does so. But if we block the adaptation mechanism by acting on the epigenome, the cell is doomed. Summing up, very few aspects concerning what we have achieved, but nevertheless, this is a great success, because we have many labs working at these aspects now. The crux of the matter that environmental changes or relational experiences with the environment may be passed down, may be transmitted, and they bring about stable phenotypes, and they bring about the development of stable phenotypes, unfortunately including diseases, and they do so through stable epigenomic changes. These epigenomic changes may be measured, and this is good news, because this will enable us to develop tools which may enable us to detect the risk for single individuals, because that is different for each individual. Another fact is that these epigenomic changes can be reverted with drugs. They are reversible by nature because they have to maintain the phenotype stable for a certain period of time. So they are reversible in the formation. And this is what we can affect with drugs. At present, we have dozens of epigenomic disease correlations here. You can have a list, you can read the list: hypertension, diabetes, obesity, schizophrenia, and bipolar disorder, Alzheimer's disease, asthma, coronary diseases. Now, we have to collect the correlations saying that there is this epigenome in the Alzheimer's disease, for example. Then we have to correlate this with a specific environmental situation. We have to decipher the mechanism and then intervene. Of course, this is going to be a long road ahead, and it can really be scaring to think of this. But what I'm telling you now would have been considered as heresy if I had said the same thing 3 years ago. So we have been taught to accept only the genetic kind of inheritance, and we have been taught to be linked to our past in all cases. But epigenomic is giving a new possibility of not being slaves of our DNA, because we are now detecting a code which does not depend on the DNA and which